So thank you very much. What I've entitled this presentation is Optimizing Photosynthesis. Hopefully you can all reflect on your school days. So some of us that's a little bit further away than others, but you can reflect on your school days and you will remember that phrase. It means making life from light. Fundamentally the biggest process, I think, I'm right in saying, on planet Earth. And I'm going to base the presentation on the work we've done at a project called Project Lannock, which is a trial site hosted and run by Agravista, 8 miles north of Northampton on some heavy soils. I'll share more about that in a minute. And we've been on a journey for 13 years, we've looking at soils, looking at grass weed control, looking at many aspects, trying to do as much as we possibly can without using chemistry. And when we started the work, we never heard of regenerative farming, and I'd certainly never heard of regenerative farming. But nevertheless, through lessons learned the hard way and the easy way, we've stumbled over and we're using quite a lot of regenerative principles. But essentially, we're having to make the system work. For farmers in a commercial situation, we don't want to fall off in yield or anything like that. And I'll share some of that work in a minute. So it's about photosynthesis, and I'll share more of the steps of that in a minute. And the data, much of it is replicated data with the strongest sort of scientific rigor to it. And so that's the journey we're on for the next hours. Lessons from Lannock. So in any regenerative system, so I'm told, we need to understand the context. And I can't emphasize, having played around with soils all my life, just how important the context is. So this is us at Project Lannock. We're on silty clay loam soils. Tricky soils. Every soil type is tricky in my experience, but they have different trickiness to it. But we're on 30% clay, so heavy, relatively heavy soils. 50% silt fraction in our soils, and sand of 20%. And we have a cation exchange, really important number to understand in your soils, which is the kind of electrical charge of our soils of 23 milliequivalents per 100 gram of soil. We've got some challenging soil chemistry, 4,500 parts per million. Those of you who understand the Albrecht system, that's a base saturation of 95% of calcium, magnesium, sort of a medium level of magnesium at 80 parts per million, and quite a low saturation of magnesium. And then we've got organic matters, although now with our different treatments, we've got a wide range of organic matters, and I'll share more of that work later on. Our 4% loss on ignition and soil organic carbon of roughly half that, or just over half of that. And then we use the soil, we use lots of indicators. With any indicators of soil or plants, if you use one on their own, you'll probably understand something, but not the full picture. It's when you start adding up different indicators when you fully understand it, or start to understand it, I should say. So we have a soil to clay ratio of 1 to 13. If you look at the Rothamsted work, I'll share more of that later. We class that as a degraded system. Our field margin, grass margin, has 40% more organic matter than our bulk of our field. We have pHs, vast range of pH is from slightly acid soils to alkaline soils, 5.5 to 7.5. And the penetrometer, which many of you may have in the back of a truck, 200 psi is what we want to achieve as deep as in soil profiles we possibly can. That's a good soil structure, or one indicator, I should say, of good soil structure. We have 200 psi at 10 centimeters, and 300 psi is about 2,500 kilopascals. And that's pretty much game over for most of the routing systems that we're involved with in an arable situation. So that's the context that we're working in. When we started the work 13 years ago now, we had significant challenges. They're not necessarily changed. We've just hopefully improved them. Some of the cropping challenges at the top there, and the fundamental one was getting on top of grass weed controls. I'm constantly reminded, David, you can have the most amazing soil health in the world, but if you can't manage grass weeds in an arable rotation, then it's slightly academic. So that's one of the major thrusts of the work we're dealing with. We have declining chemical efficacy to deal with them, and we have short rotations with the lack of diversity and some major break crop limitations. So I'll come back to that later. And then we have degraded soils, as I've already mentioned. We have a 40% loss of soil organic matter if the field margins I need to go by. We started with very low soil biology, about 4 worms per spadeful. Poor soil structure, big, angular, blocky lumps of soil that didn't infiltrate water very well. Significant levels of compaction, often erosion events. We're on a slope, and many nutrient imbalances. I'll come back to all of those later on. And then there was a lot of very intensive tillage used, lots of kind of cost attached to that, and probably a lack of attention to detail. And I don't know about you, but we kind of agree that's quite an unsustainable situation. And so that's where we started, and that's what the trial site looks like. There's a combination of fully replicated work, randomized, the strongest, fully factorial kind of scientific work going on there, linked to Nottingham University, and there's lots of systems work, and we've kind of looking at many, many different things, and I'll share some of the journey we're on. Now, one of the things we've learned in terms of photosynthesis is trying to maximize the use of photosynthesis. We call that photosynthetic use efficiency, but we've got to build it into our rotation. So one of the major important things is to think about it in rotation. So I've given you an example just over 2 years of our rotation. We kind of think of rotations as a 4-year. We struggle with break crops. But we start our rotation off, if you like, with either first wheats or with our kind of cover crop systems. I'll come back to the work of cover crops, but we often put a very low disturbance, shallow cultivation ahead of a cover crops to stimulate blackgrass control. Blackgrass germination rather, and get our cover crops off to a really good start. Early establishment, early termination, I'll come back to those in a minute, followed by a spring cereal crop. In extreme blackgrass situations, we find spring oats a very, very good, highly competitive crop in that, if we can find a good market for it. And typically we're getting yields of somewhere between, depending on moisture levels in May, somewhere between 6 and 8 tons of yield. But what that really allows us to do is not only are we doing a lot of blackgrass control, not only are we doing a lot of carbon, delivering a lot of carbon into our soils, and we now have significantly increased the level of carbon stocks in our soils, but also we're allowing ourselves to get back into the most profitable crop, which is first wheat, with an early establishment. Okay, so we're in control of the grass weeds. They're not in control of us necessarily, and we're able to get back into an early establishment. We've even got systems now established in August, and they're looking absolutely fantastic at the moment. So we're kind of missing that late establishment because we've dealt with the blackgrass predominantly in the rotation. And typically we're getting them because of the soil type, somewhere between 10 and 12 tons per hectare. So we're looking at a 2-year rotation there. We tend to look at it on a 4. We're trying to get back into first wheats, 2 out of the 4. And we're looking at somewhere between a 16, 18 tons per hectare over that kind of rotation. Okay, so that's the kind of journey we're on. That's the picture of the spring oats taken yesterday, looking really, really good. And so this is the kind of journey I'm going to be on for the next few minutes, which is looking at this kind of process of photosynthesis, looking at how we get a maximum amount of energy into our system, sunlight energy into the system. And we want to get energy in. We want to minimize the energy leaving the system. Come back to that. We're looking at how we manage air or gaseous exchanges in and out of our soil. We want air into our soil. The only place our plants can get air from is from the soil, but we've got to let carbon dioxide leave. We've got to let other volatile emissions leave our soils. We've got to let ethylene leave our soils. Very important that we kind of manage these gaseous exchanges. So we need space in our soil all connected together to enable that to happen. And then we talk about water. We've done a lot of water. We've significantly changed the hydrological aspects of our soil. I'll come back to that, but that's a really important part of photosynthesis. And then more lately, we've done a huge search on minerals, and I haven't got time to share all the details on that, but I'll share some of the ways we're starting to look at our minerals in our soil and how they deliver minerals into our plants. And hopefully at the end of that, we deliver yield, which is critically important. Obviously, we live a life, we deliver sugars, we deliver carbon into our system, and hopefully at the end of the day, we're creating a resilient, kind of profitable system. That's what we are. This is about being commercial as well as being, careful with our soils and kind of regenerative in our approach. At the end of the day, if we don't deliver a profitable, sustainable system for our farmers, then it's a kind of academic research, really. So I'll just sort of start with sunlight energy, really, and how do we maximize that. And it won't be any surprise to you that we're seeing a rapid discharge of that energy from planet Earth for a whole variety of reasons, basically. And so we can think of Earth, and I think of the whole system as an electrical system, really, as a kind of chemical battery. And its photosynthesis is our trickle charge. If those of you are familiar with your cars, then that probably is quite a helpful analogy. But unfortunately, a human life has exploited a lot of that stored energy in our soils, which is obviously our carbon in our soils. And it's kind of critical to our production systems, but it's critical to human life, and there's no surprise, but there's no plan B. And so we have to just try and create a production system over a rotation that we build more energy in than we leave. It's a net gain, if you like, of energy rather than a net loss, which I would argue where we have been for the last few decades, really. So this is the process of any energy in. Approximately on a good sunny day, we get 342 watts per square meter available to us. We want to try and harvest as much of that as we possibly can. At the end of the day, that energy, sunlight energy, is turned in a very complicated process we call the Kelvin cycles, turned into chemical energy, and eventually it ends up as shoots, which is your crop, be it a cover crop or a cash crop, your roots, and a lot of it leaks out the roots, as you know, and as exudates, and that feeds a plethora of biology, and they do a plethora of exciting things. I'll share some of that work in a moment. And ultimately, we want storage. We want energy to be stored, so what we sequester has to be more than what we lose in the system, and that's what we've managed to achieve by looking at it on a rotational basis. And our soil surface there is really important. That's the kind of interface between different biospheres. We run over it, we move it, we do all sorts of things to it, but it's a very important part of the system that we're looking at. We've been very careful with the soil surface. And really it comes back down to a kind of simple physics: energy in, the same as us as humans. We eat something, hopefully we do something, work is done, and functions are performed. And you'll see that kind of running through this. As we get energy in, carbon into our system, work is done, and things like infiltration rates and worms and all sorts of things start to perform functions for us. And so this is a continuation of my kind of electrical analogy, where our plants become our solar cells, our battery is our earth or our soil, and you can't change the size of your battery. You're given the size of the battery. So if you're heavy clay, you've got a bigger battery. If you're on a sandy soil, you've got a smaller battery. However, what you can change is the amount of storage compartments within that battery, the amount of stable carbon you're storing in that battery, the amount of energy you have in your system, essentially. And so the combination of having plants living in our soils, along with what they do to our soil biology, really changes the charge level and ultimately the amount of carbon we can store for a given soil on a given size battery. Okay, and what we wanted to reduce is the oxidation energy leaving a system. We need to minimize that as much as we possibly can. And so that's the journey on. We're trying to maximize photosynthesis. We're trying to minimize the discharge, essentially coming from tillage and oxidation. We've not managed to remove tillage, but we've done a lot of work on it. I'll share that later. And the good thing is that using cover crops, using reduced tillage in a rotation, okay, that's really important for us, because our cash crops are doing as much good as our cover crops in a sense. Then we've managed to reduce our tillage, increase our solar panels, if you like, increase our biological activity. We measure that very intensely using sort of scientific techniques, increase the amount of carbon stock, the stable carbon in our system, and reduce discharge and oxidation. So it's been quite an exciting journey for us. Just to understand that electricity in soils a little bit more, I'm not going to go in this detail, but many of you will understand base saturation and sort of the Albrecht system. Again, it's another indicator. It comes under some criticism, but it's a useful indicator. And you can see there, we're looking at the major cations within the soil, what the ideal base saturation, where we are at Lamport, which is a long way from ideal, as you can see. But the important number there is our cation exchange capacity, and that's what we've seen by increasing the storage compartment in our soil, the electrical charge, if you like, within the soil. We've seen that increase, cation exchange go up. And when you increase cation exchange capacity, lots of other things start to change in our soil. So cation exchange capacity is driven by biology, but it controls chemistry, as many things. So 1% clay, if you were able to increase your soil by 1% clay, you can't, or you might find it very difficult to anyway, will increase your cation exchange capacity by 0.5. However, if you increase your soil organic matter by 1%, okay, that will increase your cation exchange capacity by 2. Okay, cation exchange capacity is very linked to water holding capacity, as we found. It's linked to keeping nutrients in our soil, but more importantly, is what we've learned, it's now delivering more nutrients into our plants if we do tissue testing. Okay, so, and if you want to calculate it, 30% clay plus 4% organic matter, as you can see is there, 15 plus 8 gives you a cation exchange capacity of 23. Okay, so it's not difficult to give you an estimation of it. And if you times cation exchange capacity by 4.5, you're not far off what your soil nitrogen supply might be. So a cation exchange capacity of 25, just to make the maths easy, times by 4.5 will give you roughly 112 kilograms of soil nitrogen supply. So it's quite a useful number. And as I said there, it's very correlated to water holding capacity. You have to calculate it as an electrical charge in milliequivalents, and I've given you the conversion factors down there at the bottom right-hand side of the screen. Okay, but we come back to the analogy of batteries, because here we have a clay soil with a cation exchange capacity of 30, a silty clay loam, which is where we are at Lamport, 22. And if you're coming from Norfolk, so I whopped these out of a warranty bin. I can't quite get the size of the batteries to reflect the soil, but you get the kind of message. Really, it's really helpful to know that number, because that's the number that really drives so many different processes, both physical, chemistry, and biological processes in our soil. So when we're talking about getting energy into our soil, and when we're part of the rotation where we're using cover crops, and we've found cover crops to be incredibly useful and also sometimes incredibly frustrating. And the frustration bit comes from not knowing how to manage them. And we've learned that the hard way, and I'll share more of that data in a minute. But one of the key ways of managing cover crops is to get them in early. Can't overemphasize this. So this is a little study looking at many different species. I've just shown 3: black oats, clover, and cedar. They all grow really well at Lamport. We planted them in mid-August, end of August, and mid-September. Okay, and you can see the fall off in biomass. It's really quite dramatic. As day length shuts down, soil temperatures start to decline, your biomass production will go down dramatically. And biomass is super important when it comes to cover crops. Okay, we're not planting high seed rates of cover crops. Economically, that's challenging, but also we want a kind of open structure to allow black grass to come through that. So early establishment is one of the key things. What I mean by early, before the end of August, ideally. Quite tough sometimes, certain parts of the world, but that's what we're trying to achieve. So that's a little bit about getting energy into our system. I'll come more about cover crop termination in a minute. The next one is about getting air into our system. Okay, that's where our plants get our oxygen from, but also we need to see carbon dioxide leave the system, and other important gases. Ethylene is a really important one. If ethylene builds up in a plant because it can't diffuse out into the soil, okay, it just stops the plant growing dead. Okay, that's one of the symptoms of compaction. It's not mechanical impedance. It's not getting rid of ethylene that's the big problem there. So just a little understanding of how we might measure that, and we often measure, we use this term bulk density or packing density. Packing density is a little bit more real, but bulk density is the amount of soil in a given volume. It's quite a simple little calculation in the day. Packing density gives a little bit of weight from clay and silt, because we know different soils will pack down slightly differently. So we tend to use packing density now. It's a slight change in number, but it is quite important that you understand that the function of clay and silt when it comes down to packing the soil. I'll come back to that. And what we found is one of the biggest causes of infill availability. And it's not just about how much silt and clay, it's also about some of the aspects of soil chemistry in that as well. And so we've learned a lot about how we manage our soils to reduce bulk density. And this is the ideal world, quite hard to achieve sometimes. This is a bulk density around about 1.2, 1.3. Half of it wants to be stuff, sand, silt, and clay. Half of it wants space. Most of you will be more than familiar with that. And at field capacity, the space is half filled with water and half filled with air. And many things, as you can see there, affect the bulk densities of our soil, both positively and, of course, negatively as well. And there's been a recent study about degradation of our systems, and it's looked at compaction, erosion, loss of soil organic and nutrient imbalances, and this is looking at bulk densities. And for all the great stuff we're doing, moving a ton of soil every year, our bulk densities are going up in our arable areas. Okay, so we're not reducing bulk densities, unfortunately, because of loss of organic matter. We're seeing bulk density starting to gradually increase, which is not good for our production system. So now we've been able to reverse that in the work that we've done. I'll share more of that in a minute, but it's a fine line, really. That's the picture of the UK. Okay, so structure drives most of the important processes in the soil. So structure, if you had to do one thing, is super critical. And this is lots of complicated words in here, but really for us, what we've learned, both the hard way and to our great sort of joy as well, is that when plants and soil biology, when these things kind of work together, when you've got a living plant in a living soil, it creates something called a holobiont, and it creates something called an extended root phenotype, which is an evolutionary concept to say that when one and one come together, they don't make two, they make a multiplication of that. Okay, they make something outside of themselves. And what we're looking at, the thing they make outside here is better soil structure. And I think most of you will be familiar when you see a plant growing in the soil and then you dig it up. There's significantly better soil erosion generally in that than bare soil. And of course, soils are unique, pretty unique, because they have two things. One, they have structure across many different spatial scales. So if you look at it in a spadeful, you can see soil structure, or aggregation as we might want to call it. But if you look at it under a microscope, you can see it as well. It looks the same. And if you put it at the university we're connected with, Nottingham, we've got X-ray tomography machines. If you stick it in an X-ray machine, again, it looks the same. It's a fascinating medium. And actually pore space, we want pore space, different pore sizes across a wide spectrum as well to be able to manage water on systems. I'll come back to that. It's complicated to measure, but we've been able to get to the bottom of some of that. And of course, you know, when you get plants and biology and all that stuff working together, it does all these things. It's like a cartoon about how soil biology might improve the structure of our soil, because there's things that are moving, gluing, sewing, painting, all going to and eating stuff together. And if you ever listen to Carl Ritz, then he can explain that far better than I can. So this is a journey we've been on with soil structure, to our far right is where we started, sort of many years ago. And that's our control plots, really. And it's replicated, and we do 3 digs per each rep. So there's a lot of data in there. But big, blocky, angular soils, as I said to you at the start, poorly structured, poor worm counts, very low infiltration rates, and some challenging chemistry to go along with it. And it took about 3 or 4 years. But the far right-hand side of that is now the control plots, if what they look like, not actually today because they're super dry, but that's what they look like now. I wouldn't say they're highly biologically fantastic soils, but what they do is crumble in your hands. You don't need 2 hands to break them apart. So, but just by leaving them alone, okay, they've come an awful long way. I've put some sort of physical measurements there. I won't go through all of them. The next one in from the right, and this is a key message, is what we're doing is we're putting tillage into that experiment when we don't need to do it, okay? And if it's not broken, messages don't fix it. So if you're putting tillage in, and that's quite deep tillage, 25 centimeters, every single year when we don't need it, because we've got no damage there at depth, certainly, then you will denature your soil and cause damage, okay? And that's what we've done. You can see the effect there, big blocky lumps of wet, poorly natured soil, and that's causing us all sorts of problems. And significantly, we've lost, in 5 years, 17 tons measured, 96 readings down to a meter, so it's robust data, 17 tons of CO2 equivalent out of that system. So we're losing energy from the system. The middle one there is tillage plus cover crops. You can hopefully see how the cover crops have done some pretty good things there, but they've not overcome, okay, and have not built carbon. They're not able to overcome the oxidation and the loss of carbon that we're getting from tillage there. And the one in from the sort of fourth one in from the right is our 3 species of cover crops. We struggled to grow 12 or big cover crop species. We've got certain species that do very well. That's got 3 in it. And that one now, statistically, compared to the control, has captured 27 tons of CO2 equivalent. So the real difference between our tillage systems losing 17 tons and our cover crop no tillage systems, which is the second one in from the left, I'm just trying to think how you see it, is now storing 27 tons extra. So in 5 years, if you kind of make the balance between those, we've moved an awful long way with getting energy into the system. The last one is our field margin. That's still got 40% more organic matter than our best cover crop nutritional system. But we're moving in the right direction. And all of those plots within 2 to 3 meters of each other. Okay, they're all 12 by 12 meter plots in a replicated trial, and you can see dramatically how difference we're making to our soil structure. Okay, so it had some colours. Okay, so what's caused that? For me, it's just the power of photosynthesis. Okay, it's the power of getting energy into the system using plants, and other people make these comments, as well as the huge change we see in colour and structure in our soils by using that power of photosynthesis. And I like a little bit of drawing. This is kind of, this is how we are. The far right-hand one there is a control plot. And we still have a little challenge with surface compaction. I'll come back to that because we believe it's a soil chemistry thing. The next one is a subsoiling plot. Lots of damage at depth, but it gives us a nice little tilth for our plants. So it's still doing some good for us. The next one is tillage and cover crops. And we get that nice tilth, which is so valuable to us. And the final one is our no-till cover crops, but we're still getting a bit of surface compaction. So cover crops aren't a panacea of doing everything. They don't do everything for us. Okay, we still get some challenges. This is a real piece of research. It's not just, I can't just be evangelical about it all. We hit other challenges as well. I'll come back to those as well. So by adding different species in, and we've done a lot of work in pot experiments. This is not one of them, but by adding certain species in, white clover, black oats, and things like that, compile through tight soil, make an amazing difference to bulk density and improve our soil structure. Of course, we want all this connected together. We don't want nice structure here. We want it all connected together. That's the way nutrients move in our soil. That's the way plants moving together. That's really important. And what we've found, this is a little bit of science, is that our two control plots, the top left diagram there, the red one is our subsoil plots. The other one is our control plot. Of course, at every single depth, tillage is dropping our bog density. Why wouldn't it? But what we've now been able to measure, again, 96 readings, statistical finding, is that no-till cover crop plots are also dropping our bog density. You can see it visually. It's very powerful to see, but it's quite exciting to see now that the power of roots and photosynthesis is actually dropping bog density. It's quite a phenomenal thing for plants to do if you stop and think about it. And of course, that's really important because what we want is lots of space, lots of air, and that carbon that we're delivering deeper into soil profile, dropping that bog density is space for other things, important processes to take place in our soil. Okay, and this is a piece of research done by a fellow at Nottingham where they looked at columns of soil compacted to different bulk densities, 1.2 and then halfway down 1.4, which is pretty tight. It's as tight as we can compact soil by hand, really. And you can see the bottom right there is black oats, for example, which has done a really good job. And that's our experience as well, of piling through tight soil. But as they pile through tight soil, of course, they're delivering carbon and restructuring soil and doing an awful lot of really good stuff for us. And this is my own little study that I'm running at the moment, but you can see this is black oats on the right. Hopefully you can see lots of roots, and I compacted the bottom half to 1.4, the top half to 1.2. If you saw that today, you cannot see the difference between the 1.2 and 1.4. It's completely opened up that soil at depth. So that's it's quite a very visual kind of representation of what we're seeing in the soil. The other way we get air into our soil is earthworms. So we want to encourage earthworms. They're great ecosystems engineers. They improve soil porosity. You'll kind of appreciate that. I don't need to dwell on that too much. And we've seen a big change in our earthworm numbers. When we started, we were lucky to get 4 worms per spadeful. It's laborious work. Each one of those takes about a week to collect that data. But all the green ones there are cover crop systems with and without tillage. The grey ones are our controls. And hopefully you can see in almost every single year now we're seeing earthworm numbers per spadeful of north of 20, 22, sometimes even 30. It's absolutely rammed full of earthworms. So by just encouraging that process of photosynthesis, we're bringing earthworms into the system, and they're bringing lots of other benefits into our system as well. And what's their energy? This is a comparison between an NDVI of a cover crop, biomass, and earthworm numbers, and you can see energy is driven by carbon, cover crops, residue. And we terminate our crops early. A lot of people kind of say, why do you do that? Why don't you have a living root right up to the spring? We find that better because we don't get any negative effect from cover crops in the following cash crop. But that period where crops start to die, the cover crops start to die, and glyphosate takes a long time to work sometimes that time of year, is times when earthworms come up and feed on the dead residue, and we get all these earthworm channels, all these mittens on top of the soil, and that's where we believe a lot of the biology is beginning to happen. And these are pictures from others, but it always fascinates me how earthworms pile through our subsoil and bring the roots and other great benefits to the system, shall we say. Okay, we try and avoid soil compaction. A quick one, I'm not going to spend any time on soil compaction. It's a real negative effect. You can see it all over. And the way we try and avoid that, prevention is better than cure. And so we don't have soil compaction at depth anymore because we run machines with sub 6 ton axle loadings. That's a 250 horsepower tractor roughly. Sub 10 PSI tire pressures. And we use the best tire equipment we can, optimized, and that way we mitigate any damage, because damage is expensive to put in and it's very expensive to take back out again. And over cultivation weakens soils. It does lots of other negative things, but it also weakens soil. So heavier axle weight should be controlled and be on tracks if at all possible. So then we kind of looked at the role of tillage in the system, recognizing that we're probably not at the point in our soils where we can eliminate it. So we looked at this hypothesis really of what level of soil disturbance is optimum, from very heavy disturbance right down to very living disturbance. What role does living root have in the mix of all of that? And unfortunately, we get this phenomenon, and this is a phenomenon you see, and I was often blamed on no-till, but our no-tillage plots in a dry spring look as if they are on the left, much reduced plant counts, pretty much 50% down. Our conventional tillage plots, I know which one you'd rather have, is on the right and nearly double the number of plant counts. And so that's frustrating because all the soil structure looks fantastic earlier on in the season. So how have we gone about looking at that? Well, that's what our soil structure looks like in March. If we get a dry period, such as we've had this year, many of the seasons we are getting dry periods, this very tight layer, it's about 80 to 100 millimetre, forms on top of the soil. I'll explain why we think that's forming in a moment. It's not to do with machinery or traditional ways of compacting the soil. And in our tillage plots, that doesn't exist. It's very frustrating, but this is the real world situation we live in. And we can measure it using a penetrometer, very accurate. You can see the black line is our no tillage systems, much higher resistance to the penetrometer. The red one is our subsoiling systems. And we believe this is and there's much documentation about that, often seen in very dry climates. This is what we call a calcite layer. It's as a lot of moisture leaves the system through evapotranspiration, we get an accumulated hard layer caused by a lot of calcium carbonate with very high calcium carbonate, binds the sand, silt and clay, particularly the silt and clay, and causes this very tight surface compaction. We see it so often. It's very, very common. You might not even know you have it. And for us, we use a diagnostic factor of looking at the calcium-magnesium ratio earlier, and then we have to make a decision how to manage that, because once we get it, we can't undo it. Once it's in the crop, we can't suddenly just sort that out. We have to think about it prior to establishment of the crop. So this is the way we've gone about it. There's only two ways we found to resolve it. One is with rainfall. That breaks it apart, but of course we are under control of that. Irrigation is, in many parts of the world, they use to control it. For us, it's a gentle stretching and lifting of the soil. So what we found is a simple drop of 0.1, so very, very little drop of packing density in the top 100 millimeters increased our plant count by 50%. It's quite dramatic. Okay, so we've gone from there to there. Okay, it's a very small stretching of the soil. We don't pulverize it, we don't bubble it up, and we've doubled our plant counts. Okay, and we haven't lost carbon. So we have to make the system work. It's a practical system. We'd love to go to no-till completely, but we're very often, especially in dry springs, presented with this challenge. So this is what it looks like. We've keeping vertical columns of soil strength. We're not destroying the structure. When we get heavy rainfall, it doesn't kind of all pulverize, and we use very shallow 12 centimeter low disturbance cultivations. And I haven't got time to go into too much detail, but that's worked really well for us. And if you do perform a heavy tillage operation, this is another experiment we run, you will destabilize soil, and you must follow up with living roots. So on the left-hand side there, it's tillage and bare soil right throughout the winter. On the right-hand side, exactly the same soil, same year, plots next to each other. We put a very simple cover crop, and it's a dramatic difference. You would not believe it unless you see it, okay? So we're capturing that carbon leaving the soil. We're kind of capturing it in the plant and cycling it around the system. An extended phenotype effect of that is to improve the structure of our soil. And of course, the opposite to all of that is dropping packing densities too much. Okay, big, heavy machinery, heavy tillage, mechanical energy, and as I've put it, it's my words, not highly scientific. Biological energy leaves the system through oxidation and damage is done. It increases the redox. I'll come back to that in a minute. And as I said, energy leaves the system. It lowers cation. That's what we found in our heavy tillage plots. It lowers the cation exchange capacity. As you'll see in a minute, it's reducing the mineral uptake into our plant systems. Okay. And it often happens at headlands because it's overly cultivated by cultivating it several times. And also the picture on the bottom right, it's not my picture, but if you don't get your precision farming set up and you've got too much overlap between tillage, you can remove the soil structure very quickly. It's a very, very fine thing, what we've learned. And of course, you've shut down your infiltration rates very, very quickly. Okay, so over cultivation is the soil structure and infiltration. This is an overlap, and as I said to you, that's where precision technology can definitely help us. So this is where we've looked at intensive tillage. We've removed every bit of intensive tillage, any bit of deep tillage, but by going zero tillage, we run into, in dry springs, this calcite layer, this high packing density. So very shallow, low disturbance seems to optimize it for us. We don't lose the carbon out of the system, and we've taken an awful lot of costs out of our production system. So what about water? Very important in the process of photosynthesis, optimizing water. This is one of the most important characteristics of your soil. It's different for every soil type, but this is a biological phenomenon. So along the bottom is matrix potential, which is the sucking ability of plants to get moisture out of the soil. Up the side is your volumetric moisture content, and then you see the kind of... So what we want when you get a big rainfall, and it's lots of big pores to get rid of that moisture, get it into the soil as quickly as we can. But what we don't want is all those big pores on their own, because that will just drain that moisture out of that soil really quickly. We want a wide range of pore sizes, and that is created only by the combination of plants working in the soil and driving it biologically. That structure with different range of pore sizes is what we want. Little pores, medium pores, big pores to handle soil moisture. And what we're finding by doing that, by building cover crops, building our biology within our soil, we're finding now that our crops are hanging on that much longer because we've got more water holding capacity. We've also increased our organic matters, as I said, increasing cation exchange capacity. And there's a direct relationship between cation exchange capacity and water holding capacity of our soils, as you might expect. Okay, so it's about the size of your battery, but it's about how much stable compartments, how much stable carbon you've got in your batteries will drive the cation exchange capacity, ultimately driving water holding capacity as well. And we know a little bit of extra organic matter, which is energy in the system. There's many studies allows our soils to store a lot more within them. And then we've changed our infiltration rates. As I said to you at the beginning, when we started this work, the water would quickly run off. We'd struggle to get it into our soils. In the first 2 years, we made no difference whatsoever. The green ones are our cover crop systems. The grey ones are our controls without cover crops. When it's in the spring part of the rotation, it's actually in winter wheat at the moment. You can see in year 3, we started to see our cover crops changed, our infiltration rates. And then since then, they've gone exponential. Okay, and now if you have a heavy rainfall event, you walk across the cover crop plots compared to the control plots, your wellies get wet in the control plots and they're dry as anything, or as clean as anything, I should say, in the cover crops. So they're handling water, getting water into the system because of the structural changes driven by biology. And it's been quite exciting to see that. Again, that's an example of energy in physics, work done, and the function that's been performed is better infiltration rates. Okay, and that's driven by a lot of it. I think 68% of it, if you look at the R-square value, a lot of it is being driven by the amount of worms in the system. The more worms, okay, and this is why we kill off our cover crops quite early in January. We plant at the end our spring crop in the spring part of the rotation in the end of March. We've got that time for all our worms to do their magic, come to the top, create all these channels. The mittens in our cover crops things, I never measured it per square meter. But it's very, very large compared to our control plots, and that's what we believe is creating us so much infiltration rates in our soils. So the last bit of photosynthesis, and this is work in progress, so please don't think I've finished this piece of work, but we've started to unearth some quite interesting things when it comes to minerals in our soils. Just to say that in that big study, I presented it early in the presentation, you know, we certainly at Landport have got a lot of mineral imbalances. Lots of one thing and not so much of another thing. And it's not about how much you've got anything, it's about the kind of ratio, if you like, or the balances between different minerals, which is what we're finding so very, very important at Landport. And so the first thing we did was set about truly understanding soil pH, because the labs do a fantastic job of measuring soil pH, but we weren't doing such a great job of sampling. In other words, we would sample over quite a big area, bag and tag that, homogenize it, send it across to the lab. The lab would tend us the pH for that, and they'd done an absolutely amazing job for us. And I can't thank Landcrop enough for some of the support they've given us. But we found that because we didn't send enough bags, enough samples, if you like, we were getting a wide range of soil pH. So we looked at it, all the lab-based stuff, we're getting, in very short distance, we're getting pH from sort of 5 up to 8. We didn't think we were quite that strong, so this is a sampling area, nothing to do with the lab. So we looked at a pH meter, quite an expensive bit of device. We just tightened that up a little bit. But it's important you understand your pH, because, you know, if you treat it all the same and you happen to get a low pH reading because of the sampling area, and you overly apply lime to that example, we don't use lime here, we don't want any more calcium in our soils, then you can quickly cause yourself some problems in the alkaline parts of the field. So understanding pH has been a really important process, and also understanding this phenomenon. You can't measure this, and trust me, I have tried, and I got close, but it's really difficult to measure this. So this is along the bottom is pH. It's a measure of hydrogen ions or protons in a sense. You'll all understand that. Up the side is a measure of energy in and out of our system. Electrons, we call that the reduction and oxidation, or the redox scale. Okay, it's just having an appreciation of this, and what we've at Landport now, we have a real appreciation of what we're doing, because what we feel is in our big heavy tillage parts of the experiment, we are overly aerating that soil. We're creating an oxidized environment, overly oxidized. And many of our minerals become oxidized as well and unavailable or insoluble to our plants, manganese, iron. And I think most of our nitrogen becomes in the nitrate form, which is energetically quite expensive for our plants to deal with. And then actually almost in the same season, heavy rainfall event, we had 200 millimeters at the end of September last year, and suddenly those plots are waterlogged. And you come straight down the redox scale and you get compacted waterlogged soil. And many of our minerals become toxic because they become too abundant. Iron is a classic example. And what we've come to appreciate, and there's one way to, we can't measure this, but there's one way to assess it, and even the experts will agree this is the way to assess it, but we've found that photosynthesis, all comes back to photosynthesis, is the optimal way, it gets the optimum range in terms of redox is concerned. So worms, biology, living root, all the stuff that we know and love is the key way to optimize our redox. And if you want to measure it, good luck. As we tried and many experts have tried, it's pretty hard to measure. However, if you want to assess it, slightly different tone there, drop your soil in some water. I call it, there's a more scientific way, we call it a quanti-slate, but it's often called a slate test. And if your soil, as it does on the left-hand side there, this is the same field, same year, managed in a slightly different way. The one on the right is heavily tillaged. The one on the left has a very light touch to it. Okay, the one on the left, light touch. The soil is holding together. It would be there for another month, I would argue. And that, I would say, is fairly optimal when it comes to redox. The one on the right is just falling to pieces, overly oxidized. Okay, when you add water, of course, it just collapses as well. So that's a way to assess redox in your soil. Drop some of your soil in water and see what happens, basically. So when it comes to minerals, we're very, the first thing we looked at was calcium, magnesium. I think we'll all agree calcium is very responsible for flocculating our soil, which helps our gases exchange when the soils are in a kind of wet part phase of the year. Magnesium is central to the chlorophyll molecule with 4 nitrogen atoms attached to it. Very important for photosynthesis. So it's the balance between the two. We don't want to get rid of one or increase the other. We want a good balance between the two. We've not been able to achieve that. And I'll come back to that in a minute. And of course, we looked at this very historic thing called the Muddler's chart, and we looked at some of these synergistic and antagonistic relationships and the ratios between some of these minerals. And so where we've ended up, really, oops, sorry, I've missed out the critical slide. I must have hidden it up. Where we've ended up is we looked at 6 key ratios within our soil. Sorry, the slide isn't there. We looked at the calcium-magnesium ratio. Ideally, we want to get to 7:1. We cannot get anywhere close to that. We're at 40:1. And that rings alarm bells. We think, we're not entirely certain, but that rings alarm bells. For us, it's an indicator. We've got lots of silt and lots of clay in our soils, plus a high calcium-magnesium ratio. We seem to get this calcite layer as a result of that. So we've used that as a diagnostic tool to know when we might need some low disturbance tillage in our system. Okay, so for that's the diagnostic tool. We've then looked, off the top of my head, we've then looked at the potassium and magnesium ratio. We want to try and get to a 1:1 ratio of those. We know those are very antagonistic to each other. Okay, so we've got a lot of one, it may shut down the other and vice versa. So we want to get to a 1:1, and we're good at Lamport. We're definitely close to a 1:1, so it's not ringing huge alarm bells. We then looked at phosphorus and sulfur, knowing that phosphorus shuts down sulfur, and these are all sort of very documented scientific ratios. They're not just anecdotal. But we're managing sulfur anyway, so that one's not too much of a concern. We then looked at phosphorus and zinc, knowing that phosphorus will have an effect on zinc if you're over a ratio of a 10:1. And we're well below 10:1, so it's not a red flag for us. What is a red flag for us is the iron to manganese ratio. We want a less than 2:1 ratio. We've got lots of iron in our soil at Lamport, 600, 700 parts per million. And when you look at the tissue test on a manganese, we want 25 parts per million, a minimum. If you look at the science, that's where you want to be. We're well below that. We're 4 parts, 5 parts per million. So we think the iron is shutting down the manganese, as its scientific evidence seems to suggest. And the last one we looked at was calcium and boron, because the scientific evidence seems to suggest that boron is a great catalyst to get calcium in a plant. If you know about calcium, it's very mobile in a plant. It only travels through the xylem, has no membranes in the xylem, so it's a very difficult thing to get into a plant. It brings other nutrients into it, so the science tells us. And so we want a boron ratio in the soil of less than 200:1 or less than 50:1 in the tissues, and we're well below that. So for us, the red flags are manganese, boron, the calcium to magnesium ratio. Those are the three that we've kind of pulled out at the moment. So we're doing a lot more work on that. We've got a massive data set, and time doesn't allow me to share it in any more detail. But these are the kind of relationships we're looking at, soil to plant. You can see we've got lots of iron. This is iron in our soils, but we're getting about 4 parts per million in our plant, but we're not deficient. But we've got lots of manganese hanging around in our soil, but we're getting well below 25 parts per million in our plants. So that's something that's a red flag to us, basically. But when we measure the field margin, you can see organic matter drives all the cation levels in our soil. They're all up. And so organic matter is the key thing. I keep coming back to it, but the key thing in our system. And worms also are great providers, okay, of nutrition. And this is a couple of recent studies to suggest that the more worms are responsible for sort of 6.5 to 8% more grain yield, and predominantly that's because of nutrient cycling. And the cover crops hold lots of nutrition as well. I won't go into detail, but we measured a lot of this. So a lot of nutrition is in our cover crops. The question is when it's available. And for us, we've never been able to measure it in the following crop. It's the year after where that seems to come into the system. And so this is the carryover. This is after 2 years of spring cropping and cover crops to deal with blackgrass. We go into first wheats. We then split the trial no N and full N, and all the green ones are our cover crops. And where we had no N, we saw quite a 1.5 ton lift. And this is a fully replicated experiment, 48 plots. And so it would suggest that nutrition from our cover crops is appearing sort of 12 months after the use of the cover crop. So we're definitely seeing it in our rotation, but not necessarily in the crop in the same year, basically. And we run nutritional trials looking at nitrogen use efficiency. And this is a little study, and this is a little plug for the company I work for, John Deere. But one of the systems we ran was a farm standard system with a micronutrient passage. I'm convinced that micronutrient has a really significant role, and that was the one that yielded the most. And this is again a replicated experiment 3 times. So micronutrition is something we're still working on, but we're very convinced that there's a lot to be done for this. And what we've just run is a big tissue test on our winter wheat after 6 years of the tillage treatment, 120 flag leaf samples per plot, 500 per treatment, so it's pretty robust. Landcrop did the work for us. And 8 out of the 12 nutrients tested were significantly, so it's a statistical finding, because this is a replicated randomized experiment, with p-values well below 0.5, lower. So nitrogen, phosphorus, sulfur, calcium, magnesium, iron, copper, and zinc were all lower in our heavy farm standard tillage plot. So tillage is lowering our cation exchange capacity, lowering the ability for our soils to store that nutrition, but very importantly, it's now lowering the delivery of that nutrition into our plants. I'll give you 2 examples of phosphorus and copper there to lessen the busyness of the slide, shall we say. So finally, at the end of the day, hopefully that produces something, output, life, sugars, biomass yield, whatever you want to see it. One of the things it's delivered for us is carbon, as I've already shared with you. Significant way that carbon is delivered into our soil is through the exudation route. That's the one we're looking at. We're not bringing amendments into the field at Landport. We're using plants as our primary way of getting carbon into our soils. And this is the calculation. I'm not going to dwell on that too much, but you have to calculate it out properly. And we've done it very robustly by taking the organic matter, taking the depth of the profile. We've measured it 4 depths, taking a bulk density, measuring it. It's the hard bit to do. You have to take the stones out, et cetera, and that gives us carbon stock. Also, the same formula works for clay stocks, and it works for many other things as well. So you can measure the carbon to clay ratio as well. So these are the columns of soil. We break them into 4. We took 96 of these on a 32 plot trial, 8 treatments. Okay, and this is what we found. This is after 5 years. Comparing tillage to no tillage, so no cover crops involved. In the 0 to 15, we lost 17 tons of CO2 equivalent. So that's energy leaving the system. Cover crop versus no cover crop, so no tillage involved. That was a 10% loss. We gained, as I shared this before a moment ago, 27 tons of CO2 equivalent. Okay, so energy into the system. The only difference is it happened at a deeper soil profile, and that's what we missed for many years. We're only sampling from 0 to 15. Once we started sampling from 0 down to 1 meter, we started picking up these big carbon changes because the cover crops are dropping that carbon further down the soil profile. Okay, so that was an interesting result. That's an 18% gain, and the function performed of cover crops is to store more carbon. Okay, we've looked at the soil carbon to clay ratio. I won't go into too much detail, but just so you know how to calculate that, it's the soil organic matter, loss and ignition times 0.5, gives us your carbons, and then you form a simple calculation on the clay and the carbon, and you work out the ratio between the two. Simple sums at the end of the day. So this is Rothamsted's paper. I've referenced it at the bottom, and you can then see if your field or parts of your field or farm where it falls, and you can decide if you're degraded, good or very good. And we've kind of moved it from degraded to good, but our field margin is very good. So that's quite useful. There's a grassland one and an arable one as well. Okay, and so all that stuff starts to feed the biology in our soil. That output, if you like, through photosynthesis starts to do all the functions that our biology. I'm not going to go into those details. You know all this probably pretty well. My strong belief is their food source is obviously photosynthetically derived. So all of that function is coming from getting energy back into the system through the process of photosynthesis. So we've measured the biological functions. This is not, those of you scientists know, this is probably not the best way to represent this, but I did PLFA analysis, that's phospholipid fatty analysis, quite a complicated week in the lab. And I pulled out the two fungal biomarkers, which are saprophytic and mycorrhizal, and you can see all the green ones, with the exception of the grass margin, are nearly double that of our control. So we're improving things that we can see like worms, but we're also improving our microbial life with our soils, and we're improving our fungal to bacterial ratio. So we're improving much fungal activity in our soil, which is, those of you who know will know that's a pretty good thing. We're also including the amount of carbon in the total biomass of our critters in the soil. Again, an extensive piece of work, but you can see all our cover crop systems have a lot more carbon, and because of the ratio between the two, more nitrogen as well. So, you know, the output is doing great things for stored carbon, it's doing great things for our critters in the soil. We used a biometer. Those of you who come across this, it's not a highly scientifically accepted tool, but by using it, we've been able to prove the same as the scientific measures as well. So those are all our biological measurements. I'm not going through it, but we're measuring them constantly. Soil organic matter, microbial biomass, PLFA, blah, 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 blah. And you can see we're making some really good progress. It doesn't happen overnight, but it definitely comes if you persist. And the other thing we've been able to do is get on top of our blackgrass. A lot of that is to do rotation, but there's this effect, and that's our control plots, our cover crop plots on the left, our control plots on the right. If we try and break the system, the amount of blackgrass we get in our cover crop systems is significantly less than in our control system. So we're having some allelopy effect that's really, really helping us control our cover crops. And this is the data to go behind it. And so we tried to break the system by keep doing the same every year. That's not what we were about at Lamport. But you can see in year 4 we broke the system, but the cover crops in our two control plots, the grey ones there, spiraled out of control, whereas the cover crops really did a great job at suppressing our blackgrass population, basically. So just to finish off then, effect of yield and termination. So this is yield of spring crops immediately after cover crops. This is really important if you're managing cover crops. If you plant early before the end of August and terminate before the end of January, which is what we do, there is a good chance, I will not guarantee it, because we get it in some years but not others, where you might see a yield increase from using cover crops. Not guaranteed, but potentially possible. If you have late establishment into September, first week in September, a late termination, drilling on the green in March, you almost certainly will see a yield decline. We definitely see that very frequently at Lamport. And very often we have a late establishment because of the season we're in, but early termination will be a flat line. It won't increase or have a negative effect. And I can't guarantee any of that, but that's our experience based on a very long and extensive data set. So establish early, terminate early. It's a kind of bulletproof system. Outside of those guidelines, then you might have more challenges, shall we say. So in summary then, I now look at our soils and our plants through the lens of photosynthesis. Now, I'm absolutely convinced, having done 8 years of replicated science on this, collecting huge data sets, that photosynthesis drives just about everything we want it to drive. The key thing is how we manage it. Okay, it drives our roots, our microbial life, it drives structure aggregation, it drives plant health, more nutrition, as you've seen in our plant. Okay, that's what we want to do. It's kind of a new paradigm, but it does work. and it's working in a practical situation and delivering. It's not just a evangelical message. It's hardcore research done in a real field site, trying to work it out how we do it in a practical way. Okay, so that's Project Lamport. We've been able to, we feel we're in control of our grass weeds. Okay, that doesn't mean we've fixed it, but we are in control because of the rotation, because of what we're doing to our soils, etc., etc. Fantastic soil health doesn't mean you're going to control blackgrass. No way. But through rotation and doing what we're doing, we feel we're very much in control of our grass weeds rather than them being in control of us. We're building a production system that leaves more carbon behind. Okay, we're leaving more energy in the system that leaves the system really, really important, because many of the functions we've seen is driven by that energy in the system. We've removed all deep tillage. Okay, all the work we're doing now is sub 100 millimeters. That's anything deeper than 100 millimeters is deep tillage for us now, just to deal with that kind of what we call calcite layer. That's our hypothesis anyway, but that tight layer of soil in the surface. We're getting back, very importantly, to early establishment of direct drill wheat. We've even got it established in August. We'd not recommend that, but certainly well established in September without having that horrible blackgrass fear overhead. So we're back in control of the system, and we're taking a lot of cost out of the system, okay? And we haven't seen a yield fall off because of the different levers that we're able to pull to make that system work. Okay, and with that, I'm just about bang on time. I've got 5 minutes, but hopefully that's some of the lessons from Project Lamport, how we've optimized or used photosynthesis along with a few other things to really drive energy into the system, getting air and gases exchange working properly in our soils, managing water. Okay, we can't fix low rainfall, but we can manage water, managing some of the nutritional aspects, and that's kind of delivered some very sustainable, resilient yields and production for us at the end. So with that, I'll say thank you very much. If there's any questions, I'm happy to take.
Great. Thank you very much, Dr. Purdy, for a really fascinating presentation. One thing I was wanting to know, how important is plant structural diversity to the sequestering of sunlight? So if you have a diverse cover crop with, 10 different species, 20 different species, is that going to be more effective at sequestering sunlight than, say, a 6-species crop or even a monocrop? Thank you.
So really good question. So first of all, first thing we found is there are certain species that grow well and certain species that we really struggle with. So to get 12 species into our mix, we massively struggle. The other thing about 12 species is that 2 or 3 species become extremely dominant. And also comes down to planting dates. So if you can plant early, before the end of August, a lot of the small things like clovers work well for us. If you plant late, don't bother with small species because we just can't get them to grow. Okay, so that's the first thing. What we have, we've got 4 cover crop systems of different levels of diversity. And what we are finding now is the big diversity one, which for us is about 6 species, that's about the best we can achieve, has now statistically got more carbon in that soil at 15 to 30 centimetre depth than the low diversity system. And it is just functioning as a soil much better. So the answer, I don't have the exact answer, or I have a massive data set, but what for us is now delivering is more species is delivering more. But we've got to be very careful that we don't have huge biomasses, because blackgrass doesn't grow, and then it will grow in the cash crop, and we're not in control of that because of the challenge. So we've got to have that. Everything's a balance. What we've learned is everything's a balance. And we don't want to spend a big sum of money on our cover crop as well. We want to make that economic. So we have as many species as we can grow: Phacelia, buckwheat, black oats, linseed, a bursting clover, if you can get it in before the end of August. All those things kind of grow well, but we don't want a big biomass because we want our blackgrass to grow as well. But it is delivering more, statistically, it's delivering more carbon. It structures itself much better, and actually it's got a lot more cations in it as well, because we've just measured all of that. A huge bit of work, we just measured all of that. So.
Thank you very much for the talk. It was really interesting. You talked about different soil health indicators. If you have a limiting budget and you need to choose just some to check in the lab, which one you would check?
Yeah, so measuring one thing is dangerous, in our opinion. It might be right, but it might be wrong. So having a few things are really important. So with the exception of digging a hole and having a good look at soil structure, which is very, very important. Obviously, I'm teaching people to suck eggs there. For me, measuring organic matter at different depths is you almost mandatory, I think. And for me, a simple use of a penetrometer actually is also really useful. Those three things together, we've measured everything bar bit DNA because of the cost of it. But those three things, I think, armed with those, if you use them properly and know what numbers you're looking for, you could go a long way with those three things, in my opinion. Yeah, in a practical environment.
Thank you, Dr. Purdy. I share your approach to photosynthesis. I mean, without photosynthesis, the planet would just be a big barren rock. We wouldn't be here. But looking at your soils at Lamport, are you looking to improve that photosynthetic efficiency? Because it would look like your soils, with your low magnesiums and your high calciums, and I understand the Elbrecht system, you're lacking in the key, or some of the key elements for photosynthesis and chlorophyll production, such as magnesium, such as iron, because you've got low availability of iron, you've got low concentrations of magnesium. So are you doing anything to actually improve that photosynthetic ability of the crops that are growing on your soils?
So that's our latest study. So the answer is we haven't got all the answers to that, but we are most definitely on it. We have now huge data sets we have 5 or 6 years of soil analysis, tissue testing, sap testing, you name it, we've measured it. All I would say about nutrition is, for us, it's about balances. And we've been very, very careful, because if we manage the system well, we can get some pretty good yields. We're not in a situation where we've got a big problem to solve nutritionally. It would be a case of tweaking it a little bit, and that's what we're trying to look. We've got soils with very high iron, but as you rightly say, iron is not about an amount, it's about availability. That's always the case with iron. And we're starting to look at these, and I've just shared a little bit, but if I'm honest, that's a journey that we're taking the next gentle steps into. But yes, definitely, I'm absolutely convinced, as you see some of the data we ran, where we just threw a package of micronutrition at it, that definitely lifted the yield. That project, what wasn't done at project, but we definitely saw a yield benefit from that. So look, I think micronutrition is a massive opportunity, but I think you've got to be very careful that you don't upset the balances within the soil, because we know that a lot of one thing will shut down another. So I haven't got all the answers, but that's a journey we're definitely on, and it's one that fascinates me as well. Yeah.
What would you suggest if you're struggling to get on in August with your cover crops? Because we're in West and we're late.
Yeah, that's a really good question, and one we often face with ourselves. So first thing, if you're well into September, I won't say there's no value in planting a cover crop, but your growth will be dramatically reduced. So whether you get the value versus the cost is something we wouldn't do it. For example, at Lamport, we'd look at it a different way. So what we've looked at is most of the years, actually, we've been very successful getting him in sort of within the 1st or 2nd of September. So we'll go with it like that. And there's very few times we haven't achieved that. But we also looked at blowing it in. So in the standing crop, getting it blown in the standing crop, we never get quite the right level of emergence that we want, but it has been quite successful, providing you've got some soil moisture around in August. That's a big proviso, obviously. Then by blowing it into a standing crop can work really, really well. And then what we tend to do is, if we're harvesting it a few days later, we might just run over the top with a straw rake. So running a straw rake after establishment cover crops, we found a really cute little way of getting some cover crop establishment by blowing it in. So that way you get it in early. And but if you're a week or something ahead, you're not going to put a straw rake in there, obviously. But that's our kind of the way we've done it, basically.
Hi, thank you for the talk. Using SAP analysis to figure out where the stoppers are for the photosynthetic efficiency of the plants, we addressed several micronutrients, and we think we managed to increase photosynthetic efficiency, and we judge for that by measuring the sugar in the sap. Is this a proper way to understand if we improve the photosynthesis?
I think it is, yes. So I have a little plant sap meter. I think you might have seen it. I haven't brought it with me, but I have a little plant sap meter. In my mind, I'm trying to get my plant sap above 6. And I think if I'm getting somewhere close to 6 or above 6, I've not done a bad job at managing nutrition and other aspects to drive the optimum, as you call it, the photosynthesis within the plant. So for me, a sap pH meter is a great little device. It's not silly money, and that can tell you if you're doing a good job with nutrition or something like that. So what I haven't done yet, and what I will do, is to run that right across a replicated experiment, because now we're seeing some big differences in our replicated work, because it's almost a long-term piece of work now. So I haven't had time to do all of that, but early indications are that is probably a really good way of assessing plant health. Yeah. One of the better ways, if you like, without taking all the tissue analysis and all the other stuff that's possibly out there. Yeah. I think I'll probably draw it to close there because I'm over time, but thanks for listening. Hope you find that useful.